too many things to carry. I'll just leave that there. All right. Well, I really appreciate the opportunity to talk to you all in this evolutionary venue about the research that I've been doing. And what I'm going to be talking about right now is work I've been doing that I started doing way before I came to India to work at the Indian Institute of Science. And so I hope that the next time I come here, that I get to tell you about the work that's going on in my current lab. But this is work that I sort of has been carrying along with me as I move around the world. So for the last 25 years or so, I've been working on close interactions between species, mostly from an ecological point of view. And let's see, we'll go back. Okay, and so the species that I've been working on sort of span these 4 categories that are in these photographs. So, starting with the plants, I work on plants that are chemically defended with iridoid glycosides. And then I work on the butterfly species that feed on these plants. And so these butterfly species are specialized to feed on plants that are protected by iridoid glycosides, and they sequester those iridoid glycosides for their own defense as caterpillars. And then the bread and butter of my work is mostly the parasitoid wasps, and especially the parasitoid wasps that use as hosts these chemically defended caterpillars. And then the Wolbachia endosymbionts. So these are maternally inherited endosymbionts, and I work on them mostly in the context of the parasitoids, but they're also Wolbachia that are endosymbionts in the butterflies. So closely interacting species exert selection on one another, which can lead to the evolutionary diversification of one or both of the groups of taxa. And this was studied back in the 2000s by Nicholas Valberry and also Dean Bowers before that, with looking at the relationship between the plants that are sequestering iridoid glycosides and the butterflies that feed on them. And I'm going to mention their work again. And then in the 2000s, say 2005 to 2012, I and colleagues worked on the sort of looking at the patterns of diversity between the plants, I need a pointer. Looking at the patterns of diversity between the butterflies and the specialized parasitoids, and then also between the parasitoids and the plants. How's anybody doing this? Okay, and then more recently, we started looking at the relationship between the maternally inherited Wolbachia and the parasitoids. And it's when this paper that in 2024, this paper that we've been working on for a really long time came out, and then when I was asked to come and present in this venue, I thought this is a good opportunity for me to sort of glom together this 25 years of research, looking at these sort of different players in groups of taxa that are interacting very strongly. So that's what I'm trying to do. Okay. So this is the paper that sort of got me thinking, oh, I'm going to look back at these old data. So when we think about phylogenies between unrelated species kind of matching up together, the first thing we think about is coevolution. So things like an arms race between a consumer and a resource species, say in a disease or a herbivore and plants. Or we think of something like Batesian mimicry or plants and mutualists, where the evolutionary relationship between the two things drives diversity of both of the things. And then a really extreme example of sort of that kind of correspondence of phylogenies is if we think about a parasitoid wasp and its baculovirus, which that virus is integrated into the DNA, and you might even not say it's a separate thing, but anyway, it's default that the virus diversifies along with the host. But we can also have phylogenies that are changing together for other reasons, or I shouldn't say changing together, but are similarly shaped. And those similar shapes could come about if there's an already existing diversified phylogeny, and then it's colonized by some species, and those species diversify because of this new resource, and then they kind of correspond, the two phylogenies kind of correspond. Or there can be shared biogeographical processes that affect both species, and so they end up having similar phylogenies, but they're not even interacting at all. Okay, so the checkerspot butterflies are in the tribe Melatiinae, and the ones I'm focusing are the genus Euphydrius and the genus Melatia, which are, this is the tribe, and these are the two genera, and these guys have in common, sorry, is this not working very well? Okay. And these guys have in common that they eat the iridoid glycoside producing plants. And the iridoid glycoside producing plants are in the subclass Asteridae. So these are plant families in the subclass Asteridae. And then these ones that I've highlighted with the green are ones that produce iridoid glycosides. And if you look at these checkerspot butterflies, they feed primarily on plants that are in the iridoid glycoside families, and within those families, they only focus on the iridoid glycoside producing plants. And back in the 80s and again in the 2000s, it was concluded that the plants had diversified, and then after that, the butterflies had colonized them, and then they had diversified. And so the diversity in this sense is driven by the pre-existing plant diversity. So this is already known. Okay, so now before I get to the parasitoids, I just want to remind us what are parasitoids. So parasitoids are insects which are parasites of other arthropods, and they lay their eggs in or on an arthropod, and then they grow up using that host, and the host dies, and the wasps, usually wasps, are free living as adults. And something to keep in mind is that all Lepidoptera and pretty much all insects host a whole community of parasitoids. So parasitoids are kind of small, but there are gazillions of them. It must be that it's the most diverse group of insects. And parasitoids kill a lot of their hosts. So just as one simple example, where I worked in Finland on the Glanville fritillary butterfly, by chance there's no egg parasitoids, which is unusual, but at least 30% of all of the caterpillars are killed by parasitoids, and then of the remaining, 20 to 50% of the pupae are killed by parasitoids. So this means that mortality by parasitoids is huge, and this example is not an exception at all. This is completely normal. Okay, so parasitoids are in the order Hymenoptera, which includes sawflies, which nobody ever thinks about, but it also includes bees, predatory wasps, ants, and then the parasitoids. And the parasitoids that I'm going to be talking about are in the family Baconidae. And this is the genus Cotesia, which is a huge genus of parasitoids that's cosmopolitan. There's maybe 2,500 species. Who knows how many species there are? There's less than 500 of them have been named. There's quite a lot of species. And most of them are endoparasitoids of externally feeding Lepidoptera, butterflies and moths. So many checkerspot butterflies co-occur in meadows, and they feed on the same plant species. They look really similar. They have similar biology. And the Cotesia parasitoids using the checkerspot also co-occur in the same meadows, and they look very similar to each other. And morphologically, they're so similar that we think of them as being in cryptic species groups, meaning we can't tell the species apart morphologically. So the question is, what's the evolutionary relationship among the wasps in this specialized system and the association, and its association with the butterfly phylogeny? So a while back, we collected as many of the parasitoids as we can from the different butterfly hosts, and we made this phylogeny. And I'm going to show it to you in two ways. So what we see here is that, well, for one thing, at that time we found that there was 15 rather than 6 species, and then since then we found several more species. And they mostly fall into 2 clusters, the Acuminata cluster and the Melitarum cluster. And so we call these the Acuminata and the Melitarum cryptic species groups, because if you look morphologically, they just look so alike, and their lifestyle is so alike. Okay, then you wonder, okay, where do those 2 groups of Cotesia fit within the Cotesia, within the whole genus Cotesia? So then we sequenced more and also got sequences from more places of more Cotesia. So this is just showing the whole Cotesia genome. And what we see here is that the Acuminata group and the Melitarum group are not very close to each other. That the Acuminata group is exclusive to the checkerspot butterflies, and the Melitarum group is not exclusive to the checkerspot butterflies. There's some moth hosts that are not related at all. And then there's a North American one, which is quite distantly related. Okay, so these clusters are spread throughout the genus Cotesia. Okay, so then this is just graphically representing our question, what's the parasitoid phylogeny relationship to the host phylogeny? And so these are the 2 cryptic species groups, and these are are 18 of the butterfly species, and I spent gazillions of hours staring at this in various different forms. So the parasitoids have 1 to 3 host species, mostly 1. If there is multiple host use, then there's examples where they're both related and unrelated, or closely related and not closely related. And where there's 2 Cotesias on a host, the Cotesia are from different clades. So even in one meadow, you can have the 2 different clades of Cotesia on one host. Okay, but all in all, this is not showing any kind of phylogenetic mirroring at all within the group. Okay, just to be thorough, we said, okay, fine, they're not really sorting themselves out among the butterflies, maybe they're sorting themselves out among the plant species. So the parasitoids directly use plants because they use plant cues in order to find the host, and they're affected by the host plant chemistry. So they have their own personal relationship with the plants that's not through the butterfly. So again, this is just a graphical way to represent this. This is the different parasitoid species, the 2 different cryptic groups, and then this is different plant families, different plant, species within the different families, and lines going all over the place, which, so one thing is obvious, which it has to be this way, is that wasps that use more different butterfly species also use more plants. That has to be true because the butterflies are using different plants. And then also that the wasp doesn't seem to segregate in any way among the plants. Okay, so this will get summarized. So anyway, so now we're moving on to the Wolbachia and the Cotesia. So Wolbachia is a maternally inherited endosymbiont. And so these kinds of endosymbionts can do all sorts of strange things in the host. But one really common thing is one way or another, they induce a pre-mating or post-mating reproductive isolation. For example, if they cause cytoplasmic incompatibility between infected and uninfected individuals, then that can mean that only infected can mate with infected and uninfected can mate with uninfected. And this can be And so this kind of reproductive isolation can cause speciation of the host. So we were wondering, oh, maybe that's happening in our Cotesia. We sort of didn't see anything compelling showing that the host butterflies were sort of explaining the specific diversity of the parasitoids, so we thought, okay, maybe it's the endosymbiont instead. So that was the motivation for this. So we characterized the infected status from 21 different European Cotesia species that were using these hosts, and we looked at their phylogenetic correspondence. Okay, this is an awful picture, but okay, so this is the parasitoids, this is the Melipterum group, and this is the Acuminata group. And then this is the Wolbachia. Wolbachia is generally split into subgroup A and subgroup B. Most of ours are in subgroup B, but not all of them. But you can see within the phylogeny for showing of the Wolbachia that, again, there's no particular pattern. The Wolbachia that are in the different Cotesia species are not especially closely related in the big picture or even in the small picture. So there are a few examples in here, if you look close enough, that were closely related. Cotesia have very similar Wolbachia strains, but for the most part it shows a pattern of horizontal transfer between the phylogenetically distant lineages of hosts. Okay, so horizontal so Wolbachia is maternally inherited, meaning the mother passes it on to her offspring. So we can think, well, if it's maternally inherited, how would you ever end up getting spread between populations? We would think it would be more like that virus example that I showed you. But actually, it's maternally inherited, but in this case, it's actually, when the eggs are laid, it's like on the outside of the eggs. And there's various ways in which you could, through sort of contamination, you could get horizontal transfer. And so if you're curious about sort of mechanisms of horizontal transfer, this is a paper that will be coming out soon, hopefully, where we look at what are the possible points at which horizontal transfer might occur. So successful horizontal transfer of Wolbachia is extremely rare. Wolbachia isn't like a disease where it wants to get from one host to the next. Wolbachia is built all for horizontal transfer, yet we know from looking at the different phylogenies that it has to happen some of the time. And so in this system, there's sort of two places in which it could be happening. One of them is between the Wolbachia from the butterfly and the parasitoids getting mixed up during unsuccessful parasitism. So they could share Wolbachia in that intimate process of being parasitized. Also, two different parasitoid species could share Wolbachia in those situations because there's a good fraction of the parasitism events are unsuccessful. So there could be Wolbachia shared between hosts and parasitoids. And then occasionally, there could also be Wolbachia potentially picked up from the plants, which it would somehow get dripped onto, things like this. So these very small chance events must somehow lead to these sort of big phylogenetic host jumps that the Wolbachia must be making. And so I guess the current work that is happening in this is towards the end of understanding, trying to understand mechanisms of horizontal transfer, which are very rare events but must occur. Okay, so Meliptea nuphidius butterfly diversified on plants that were already making iridoid glycosides. That's something we already knew. The two parasitoid wasp cryptic species groups colonized the butterflies and presumably then also diversified themselves. The Wolbachia and the Cotesia don't show any evidence of the Wolbachia facilitating speciation for the Cotesia in this case. And so, while I still think, species interactions likely lead to diversification, it doesn't, in this very system, there isn't really evidence of coevolution, even in the case involving Wolbachia that's maternally inherited, which is the most likely one that you would see it in this setting. And I guess, so this is obviously work over many, many years, so there's a lot of different people. The butterfly-parasitoid work, Maria Cancare was a PhD student working with me, and Ilka Hansky and Konstantis Stefanescu. Parasitoid molecular work, more recent Wolbachia work, and funding mostly from the Academic Academy of Finland. And that's it. I'll take questions. So that goes against my assumption that the Wolbachia would be causing speciation. And because Wolbachia wouldn't be in the lineage long enough to cause that. And I don't think it was, I maybe misspoke, I don't think it was assumption, it was just sort of the background thing that we wanted to, had in the back of our mind, maybe this is occurring. These Cotesia have not diverged so long ago, so it's not impossible. But definitely, yeah. We did not find any evidence of that.
Are there any other questions?
Hi. Hi. So I was quite taken by this contrast of what you have studied here in Europe to Astraptus parasitoids in Central America, where they have shown that there are close to 10 species of parasitoids which are highly specialized compared to what used to be known as one very generalist species.
Yes.
And you mentioned that there may be something like this going on in these species.
Yes.
Has anybody done any detailed phylogenetic work to see how many species there really are?
In this one?
And then also genetic work to see whether there are specific types which occur only in specific butterflies?
Well, yeah. So, we have sampled pretty in the European Melatia and Euphydrius. We have sampled all the things. We have sampled from many places, and so I think we have an okay. There's probably something else out there, like we found some totally new species. But I think the sample we have is okay, and we've done, and for all of the things that we're calling species within the cryptic species groups, we've done behavioral work to see that they're different. So we've done mating behavior and also so they have to be willing to mate with each other, and they have to be successfully parasitize something in order to sort of know the host and know who's a species. And so in that sense, among these ones, I think we know the actual picture of what's going on. And it doesn't seem to be the same as the sort of radiation of closely species. I mean, it's of like these co-phylogenies. It's more diverse, or more diffuse. So they live together in the same kinds of environments, often in the same actual meadow, and there's some overlap in host use, and it's just a kind of a messy, diffuse evolutionary relationship.
The thing is that in a lot of these species, the cryptic species are so difficult to identify just by visual inspection that genetic analysis may be necessary. So it may be good to do that at some point, is all I'm suggesting.
Okay, I can show you. We have actually quite a lot of genetic analysis of the I can show you it, of the parasitoid species.